I'm Xiongtao, a PhD student in Murphy Group, and now I'm going to talk about a model for modeling cell and nuclear shape. It's called diffeomorphic model. So there are a lot of different models for modeling cell and nuclear shape. Basically, we have two types: parametric and non-parametric models. So Bob talked about some parametric models before. Basically, in this kind of model, we assume some specific kind of model, and the model contains specific number of parameters. And it includes outline model, ratio metric model. I will talk them a bit later. And for non-parametric model, it will not assume specific number of parameters. So it can have some advantages that it can model more general cases. So diffeomorphic model is a kind of such model. And so now let's go over some parametric model to show why we would like a non-parametric model. So here are some images of the cells. We can extract boundary of the cells, and then we use the coordinates, x, y coordinates of the boundary, use the features, and we can perform principal component analysis. So in the later panel, it shows the results of principal components. So basically, we can see the first mode can capture most of the variance, and the second mode can capture larger major variance. So these two modes can capture more than 90% of the variance of the shapes. And we can use the results to build a shape space. A shape space is a vector space that in each point of this space, it can represent some shape. So here we use the first two modes of the results to build a shape space. The x-axis represents the first mode, and y-axis represents the second mode. So from here we can see for the first mode, it most captures the size of the cell. From the left to right, the size gets larger and larger. And for the second mode, it mostly captures the roundness of the cell. So with these two modes, we can for each point in this space, it's a combination of the cell with specific size and also specific roundness.
Yeah, do you have any idea what features the first principal component was capturing? Or the second?
Yeah, as I said, the first component captures the size.
Or, okay, I mean which size in general, or any features of the size in particular?
No.
Just the overall.
Yeah.
Overall volume, or area, or surface area.
Yeah. And yeah, this model is very easy. It's do not assume a lot of parameters, only the mean shape and the variance. So, but sometimes if we want to model very general cases, in the shape space, it can represent some very unrealistic shape. So for example, the shapes are in the circle. It may have some self intersection between the boundary points, which is not acceptable for us to model cell shape, because in cell shape, we don't have such self-intersections. And another model is the ratio model, as Bob talked a bit about it. So basically, we model the cell and nuclear shape jointly, and we calculate the ratio of the distance between the cell boundary, nuclear boundary to the centroid of a nuclear shape, and we rotate the line to get the ratio maybe every angle, and we can get 360 ratios. And we use these ratios as a feature, and we perform principal component analysis. And for 2D, we can keep the first 10 principal components, and for 3D, we can keep the first 25 principal components. Yeah, this model is also very simple, but unfortunately, sometimes the CL will not satisfy the assumption of this model, because if we want to model this CL, the PC12, it has very long neurons. And if we want to try to extract the ratio, if we suppose this is the centroid, if we draw a line from here to here, then we may get multiple intersection points with the CL boundary. So we have, but in this model, it only assumes a single intersection point. So we have to choose one. Maybe if we choose this one, then we lose the information of this non-neurons. So here is an example reconstruction of the CL shape from this model. We can see it do not capture the whole morphology of the CL shape. It only mostly captures the shape of the CL body, but not neurons. So in sum, here we looked at two types of parametric model, and we found some problems. The first problem is they are too simple to model some complex cases, and the other is they can our CL shape may be very complicated, and they do not satisfy the assumptions of the specific models. So, and the fundamental reason is that these models, they just rely on some very limited number of parameters, but which determines that they cannot model very complicated data. So this is the reason why we would like to turn to some non-parametric model, where they do not assume specific number of parameters or specific assumption of the models. And the diffeomorphic model is such a model. And now let's start the principle of diffeomorphic model. Diffeomorphic model depends on an algorithm called large deformation diffeomorphic metric mapping algorithm. So the algorithm rely on a mathematical concept, diffeomorphism, so which is a smooth and invertible mapping from one coordinate system to another. So here is an example. Suppose here's a diffeomorphic mapping from the regular rectangle grids. So the original one in this grid, these are straight lines, but we can wrap the straight lines to the smooth curves, and they are one-to-one mapping. In this case, we can move the pixels in this grid to specific locations. So here's a more concrete example. So we can map the continent in the globe. Here, this is the 3D to our 2D map like this. And we can also map this 2D map back to the 3D Earth. And we can use the same idea to do the diffeomorphic mapping for the images. So suppose here are two faces. We want to map the left one to the right one. And yeah, when we observe these two faces, we can see their mouths are different a lot. So here we can wrap the pixels around the mouth, so that for here we can push it here to here, and then the middle to the bottom, so that we can get a wrapped mesh grid, which represents the deformation field we use to map this face to the other one. And with the same idea, we can apply this method to the cell or nuclear shapes. So here in the upper panel, we use the binary image to represent the nuclear shape. And then we can do the same thing for different nuclear shape. We can just wrap the pixels and move them to specific locations so that they can match together. And to make it clear, it is not just some kind of interpolation. As we so suppose there are two phases, we can see an example here. So if we just do linear compilation of these two phases, if we want to match these two phases, then we will get a result like this, which is not a good mapping of the two phases.
I guess, but for the shapes, you're assuming it's a continuous. It's continuous, right? As in, whereas on the bottom, you're not making those assumptions.
Oh, you mean here?
Left and the right nuclear shape.
Yeah, here just showed 4 different nuclear shape. Later, yeah, we will talk about how to do evolution from one shape to another.
I guess I'm just questioning. You're saying you cannot interpolate the images, images if they're vectors. I'm wondering why that's true for the real nuclear shapes.
No, it's not true.
Yeah,
Oh, sorry.
He's just showing these images.
Yeah, here it just show the image and
And to go from one end to the other,
yeah.
you could do it. You can't do it using a linear interpolation.
Yeah, you can do it, but it's will not get a good result.
Illustrate that
Yeah.
he's showing these two images here, where he's showing
Right,
what the
saying that-
interpolation would be.
Right, I'm
And
saying
in
is that
between, you don't
okay.
get.
All right, that's fine.
Up above, he's not showing what the interpolation would be on the intermediates. He's showing these are the shapes that you want to have progressed with each other.
Yeah.
Yeah, we're just saying linear interpolations aren't necessarily good, but some sort of non-linear interpolation would be better, as seen on the top.
Yeah.
That's not any kind of non-interpolation on the top, though. Those are real.
Those are real images. He's not showing that those are interpolations.
Yeah.
He's got two different concepts here. on this slide.
Yeah. So if we if we use diffeomorphic method to match these two phases, then it will see how difference of these two shapes, two faces. So the face of lady is smaller, and the face of cheetah is larger. So it will stretch the face of the lady to make it larger, and also compress the face of cheetah to make it smaller, and also move the eye, nose, and mouth to specific locations, so that if we just after that the transformation, we can just add them so that they can match very good. It's much better than just linear interpolation as we saw before. So here we need to know the diffeomorphic mapping is a nonlinear transform. It's not just a linear interpolation between the shapes. And the LDDM algorithm is an iterative algorithm that it will, in every iteration, it will change the shape a bit, and finally, it will change to the target shape. So from shape A, and we change the shape a bit, and more, and then go to the target shape. And in every iteration, we can assume we need to do some work to change the shape. And then we can define our quantity distance as the total work that we need to do across all the iterations. And this quantity can also represent the dissimilarity between two shapes. That is, if the two shapes they are quite different, this means we need a lot of work to morph one shape to another. This means the distance is large. And if the shapes are similar, this means we don't need much work to do. So the distance is small. So here, this quantity, in sum, this quantity can measure the dissimilarity between two shapes. And also, again, I want to make it clear, it is not a linear interpolation or linear process between two shapes. It's like it's a nonlinear transform. This is similar to if we want to travel from a city to a very far away city on the Earth, if we just follow, we need to travel on the surface of the Earth, which is a curve instead of a straight line. So here is the same idea. It's not travel through a straight line, but through a curve. So it's a nonlinear process. So here is an example of the diffeomorphic mapping from the left shape to the right one. These are the 3D hierarchical with both sphere and nuclear shape, and we project them to 2D for visualization purpose. So from this one, and then we can evolve to the target shape like this. And now I have introduced the basic principle of diffeomorphic model, between the diffeomorphic distance between two shapes. And but what if we have a lot of cells? Then we can compute the pairwise distance between them to measure the dissimilarity between these shapes. And if we have n cells, then we will get a distance, the n square distance between them, which can form a distance matrix. But we want to find a map that we can put the cells in the map so that their distance are preserved. So it's just like we have a distance matrix between some cities in the United States, and we can use some method to put them to a coordinate system like this. So, and they are in the relative right position. So, such as the Boston is in the northeast, Miami is in the southeast, and Seattle is in the northwest. So we can do the same thing for our cells. We can use a method called multidimensional scanning to project the distance matrix to some coordinate system. And for each cell, we use a coordinate to represent the location of the cell in the space, and such that their pairwise distance are approximately preserved. So here is an example of the shape space we built joint for both cell and nuclear shape. And we can see that it can represent the morphology difference between the cell groups. So for this group of cells, we can see they are mostly large and round cell, where for the cells here, they are small and not that round. So now I have talked about the principle of shape space model, that we compute the distance between them and build a shape space from the distance. And in cell organizer, there are two modules, the training and the synthesis. So now I'm going to talk about how to train a diffeomorphic model in cell organizer. So here is the basic pipeline. I think you should have a basic understanding of this. So first we compute the pairwise distance between each pair of cells. Then we put the distance to our distance matrix. And after we fill the distance matrix, we can use multidimensional scanning to project them to our coordinate system, which is a shape space. And in this method, it needs 4 distance matrix. That is, we need to compute all pairwise distance. It's in the order of n square of the cell number, and it would take a long time to fill the whole distance matrix. So we can use another strategy. That is, every time we just pick some number of a small number of cells, and then we finish the distance matrix between these cells. And after we do that, then we can add a new cell and compute this column of distance So we can get a distance matrix 3 by 3. And every time we add a new cell, this is 4 by 4 and 5 by 5. And in this process, because the distance matrix is symmetric, so every time we just need to compute each column of the distance if we add a new cell. And if we don't have time or we want to see the results, we can just use this 5 cell, the distance matrix of this 5 cell, and build a shape space of these 5 cells. And later, if we want to compute more, we can just repeat the same process, so that we can get a larger distance matrix and build a shape space of more cells. So, however, these two methods both assume we need the full distance matrix between the cells we want to use for the shape space. And it may take a very long time. So for about 250 cells, we may even need about 10 days, which is a long time. So we can use a lot strategy that we don't need to compute the full distance matrix. We can just choose some cells as landmarks and compute the distance between all cells to these cells. So if we choose the first 3 cells as landmarks, then we just compute the distance fill the distance matrix of the first column, second column, and third column. And then we don't compute the distance between all other cells. So then we can use an approximate multidimensional scaling method to build a shape space. And in practice, we found the shape space using this method is also good compared to the shape space of the full distance matrix. So now I have introduced several different strategies for building the diffeomorphic model. Now I'm going to talk about the synthesis in using the diffeomorphic model. So as we said, we can also generate new shapes from our model. So the synthesis is for this purpose. So once we get our shape space, which is the model, we can also synthesize some new shapes. So suppose we want to synthesize the shape in this location, which is not seen from our data set. So we can just using some surrounding cells, and we could do a pairwise synthesis. As I said before, in between these two cells, they can represent the evolution between these two cells. So we can first synthesize a shape in between of these two cells, and use this cell with another cell to synthesize their shape, and finally we can synthesize the shape in the target location. So this is the basic idea for synthesis. And now let's talk about the application of the diffeomorphic model. The first application is to model the distribution of the shapes. One method is to use the nonparametric density estimation. So suppose we want to load the probability density in this location. We can do this by contrast the polyhedron in the shape space between the nearest cells, and then we can model the probability density as 1 over the volume in each polyhedron. This is one method. And another method is to use some parametric density estimation methods. Here we can use Gaussian mixture model. So here we show the example of a model of modeling the distribution use different number of Gaussians. And we can pick the optimal number, use some criteria such as AIC, BIC, and so on. So, as we talked before, in the shape space, in every position, it can represent a shape, and also in our algorithm, it captures the most variance, like the multidimensional scaling. It can capture the most variance of the shapes. Here it's like PCA captures the highest eigenvalues. So it can be used as normal feature space that we can use the coordinates of the shape in the shape space as features, and we can perform classification or clustering tasks using the features. And here is another application is to study the relationship between some protein or DNA intensity. So here we want to see which if there are any relationship between the morphology and the DNA intensity, and we calculate the correlation between each components with the DNA intensity, and we found the third component in the shape space is have very high correlation with the DNA intensity. This also suggests the DNA intensity may affect the morphology of the cell shapes of cells. So this leads to another biological question. We want to see if there are any dependency relationship between cell and nuclear shape. So we can answer this question by building, by prediction from nuclear shape to cell shape and from cell shape to nuclear shape. So we can construct the cell and nuclear shape separately, and then for each nuclear shape, we can predict our position in cell shape space, and by interpolating the same the distance from the cell shape of its neighbors, and then we can find our predict position in nuclear shape, and then we can synthesize their shape, and we can measure the prediction error as the distance between the actual shape and the predicted shape. And here are the prediction results. That the first, the upper panel is a prediction from nuclear shape to cell shape, and the lower panel are the prediction from cell shape to nuclear shape. So the color stands for the prediction error. The bluer, the smaller the errors are. So we can see that for both cases, most predictions are quite well. They have very small prediction error. This indicates there are some dependency relationship between the cell and the nuclear shape. Another application is to visualize the different cell types. So here are the H1299 cells with different genetic modification. Each color represents one modification. So here, from the shape space, we can see that for the same type of cells, they mostly clustered together. And there's another information here that for some type of cells within the same type, they have some heterogeneity between the shapes, especially for the blue and the yellow ones. The last application is to apply the method to drug screening tasks. So here we have some images with the treatment of cells for about 25 drugs, and these drugs come from 5 mechanisms. Some drugs work on DNA, some drugs work on microtubule. So, and these images are from our paper, our PNS paper. They use one method to extract feature and do the same thing. So our question is, could we use the features from diffeomorphic model to help the prediction of the mechanisms of the drugs? So the basic method is to extract features of the cells. We here for the diffeomorphic model, we use the coordinates in the shape space, and then for each, then we cluster cells to K clusters, and then we can compute the probability density of cells in each cluster for each drug. Then for each drug, the feature is the probability distribution of the clusters, and we can use the feature to classify the drugs. So here is the shape space we built. Here the color just means different cells. We don't assign them to different drugs or something else. And here is the drug prediction results. So we use a linear combination of the feature from their paper and the feature from diffeomorphic model. Here 0 means only use the feature from their paper, and here we use only diffeomorphic feature, and here are the linear combination between the features. And here the dashed line here means the p-value of 0.05, which is if they are above this line, this means the prediction is significant. And we do this for different number of clusters. So if we just compare the results of their feature and our feature, we can see for most cluster number, we can predict 3 classes, where they can only predict 2 classes, 2 mechanisms. And if we combine these 2 features, we can get a better prediction for the mechanism of microtubule, which are better than both of these 2. So this means our diffeomorphic feature is better than their feature, and if we combine these 2 features, it can help the prediction of the mechanisms. So in sum, I have talked about the principle of diffeomorphic model, that we use Lvds algorithm to compute the distance between cells, and then we build a shape space using the distances. And for the training, basically we have 2 strategies. The first strategy is to compute the whole distance matrix, and the second strategy is to just use some cells as a landmark and compute the distance to these landmarks. For the synthesis, we use the surrounding cell to sample the shape in target locations, and the model has a lot of different applications, such as visualization, or cell shape related problems, cell dynamics, and drug screening, and so on. Yeah, that's all. That's all. Do you have any questions?
So you say the Lvds algorithm is what's used to compute the distances in shape space between cells, and I think I didn't quite grasp how that distance is generated. What is it? Is it looking at computational effort required to make that transition between 2 cells, between 2 image shapes?
Yeah, here is the definition of distance. As I said, it is an iterative algorithm. So in every step, it would make some efforts to change the shape, right? And then we define the distance as the total amount of work we need to do across all the iterations. So it's
work is defined how there? Work is
yeah, it has some
generating heat or what?
Yeah, here because it has some mathematical definition, but here we just Yeah, to make it simple, we just say this amount of work. In the, yeah.
Sorry, can you clarify what each iteration is doing here exactly? What is the work being done?
Oh.
Like how is iteration 1 transformed to iteration 2?
Yeah, it uses, basically in the algorithm, it uses maybe the gradient descent. So in every time, it will have some gradient and change the shape a bit. And the quantity here,
I guess like what's the objective function? What's the objective function?
Yeah, it's a bit complicated. You can refer to the original paper for the definition of the objective function. Oh yeah, we can. Yeah, here for visualization purpose, we only show a shape space of 2D. In practice, we can build a shape space for higher dimension. 3D. Yeah, in default, we use 7 dimension in cell organizer.
So it's a 2D big enough for the predicting power of your applications?
Usually we don't use just 2D. We use usually we use 7D or higher dimension because it can capture more variance of the cell shapes.
7?
Yes.
How many stacks do you use then? It's licensing to these stacks.
Actually, this is actually, as I said, this is a non-parametric model. So it do not assume specific maybe stack or something to the shapes. We can input arbitrary shape. Maybe some cell have 2 stack or some have 3 stack. It doesn't matter. It can use this algorithm. It can do evolution to match the 2.
Right. But you said you don't use 2D. You have.
Oh, we can. Yeah. For the question Xin asked is for the shape space. But for this algorithm, we can use both 2D or 3D shapes. It's very generic, so.
Yeah, so I guess it's sort of the question I was trying to ask earlier, but for each of those 7 PCA components, for example, you said for the component 3, it was similar to, or it mapped to DNA intensity to some, 0.55 R squared. Do we have other ideas for what the other components might be mapped to?
No. This work is work from our previous members, so it's not my work. I don't know the technical details, but here we found this third component. It's
basically choose the parameters for the component, or you can, or like.
Yeah, we can compare the DNA intensity with any number of components, but
right.
we found the third one has
right,
very strong. Yeah. Yeah, this is similar to the shape space we saw in the outline model. It's just because.
Will they choose parameters that have purposely choose parameters that have high variance right across?
No, this is, as I said, it's a non-parametric model. So we don't choose any specific parameters. It's automatically generated by the algorithm, and then we found it has a high correlation. Yeah.
Right.
Yeah, this model it choose specific parameters and it can capture the variance with different meanings. Yeah, also yeah.
How sensitive is this to? So you're using immunofluorescence data. With cells and culture, so usually there's pretty, you can basically threshold it so that pretty easily, so that there's no background, essentially.
Yeah, here we use just binary image for the shape because here we only start the shape. We don't consider the intensity in the cell. And if you use for the segmentation, maybe you can choose different methods. If the shape do not change a lot, then it's not very sensitive to this threshold in segmentation. Yeah.
Right. I'm just thinking about like trying to apply this to tissue where there's an extracellular space that may have some level of staining.
Oh, yeah.
Have you explored alternatives to PCA to generate the shape space?
Hmm?
Have you explored other non-parametric methods to generate shape space? PCA?
No. Okay, maybe we can have a rest, and then I will talk about the protein model for. Maybe I can start to talk about the modeling of protein distribution in cell glyzer. So here, we apply the model to analyze the T-cell dynamics in TCL, and we publish it as a paper last year. So also, I will mainly focus on to talk about the pipeline, how we analyze the T-cell dynamics, and in the meantime, we can know how could we build the model. So first, the background of TCL. TCL is a kind of immune cell. It will bind to antigen-presenting cell to trigger its stimulation, and during this process, the actin and its regulators will be recruited to the interaction region. We define the interaction region also called the synapse, but it's not the same definition of synapse in neuron. Yeah. And there are a lot of actin regulators. So some may work in this process, some not. So the question is, we want to find the regulators that are involved in this process. So this is a collaboration work with Dr. Christopher Woodring in the University of Bristol, and they took the images of the TCLs. We have DSA and GLP labeled 3D time series images. So we labeled a lot of different proteins with GLP and image them. And here we analyze the 9 proteins acting and its candidate regulators. So here are the images, the example of the images. Here is our basic pipeline for building the models. So first we segment the cells, then we align the cells so that make them the orientation uniform. Then we unify their shape so that we can compare them. And in the last step, we build the protein distribution from the uniform shape. So now I'm going to talk about the detail of the pipeline. So before the pipeline, we need another input besides the images. We need to annotate cell couples. So there would be a lot of different cells in a field when we image them, but we only choose some interested cell couples, and we also record the time at which the synapse forms and also the correlates of the synapse. There are 2 kinds of annotation methods. Either we record the center of synapse or the 2 sides. Basically, the later method is better because it contains more information of the orientation of the cells. After we get the synapse, we can locate the cell couple, so we can crop the cell so that we only keep the interested T cell. And then the next step is to segment because there are some backgrounds. Unfortunately, a lot of our images are with no contrast. That's the traditional segmentation method may fail for our images. So we use a 2-stage snake segmentation method. So in the first stage, we choose an initial control based on the synapse location, and we involve them to find the approximate. Oh, sorry. This should be a movie, but I think there's something wrong with the PowerPoint, so we cannot show it. So this is the first stage. We find the approximate contour that represents the boundary of the cells. And in the second stage, we use some more strict parameter and use the output of the first stage as the initial contour to refine the segmentation. So at the end, we can get quite good segmentation for most cells, even they have low contrast. And after we segment the cells, as I said, cells, they have different orientation in the field. So to compare them, we need to make their orientation uniform. And we do so by rigid transformation, that we do rotation and translation so that we can make the synapse region. This is the upper is the synapse to make the synapse region facing up for all cells. And also we rescale the cells so that their volume are the same. And this we choose the volume with using our template, a half ellipsoid. This is I will mention it later. But after even we make their size the same, their orientation the same, they still have different shapes. It's not easy to compare different cells. So here we use a long-rigid alignment that we want to make their shape uniform. So we do so by the diffeomorphic model, as I talked before, use the Lvds algorithm. We match the cell to the uniform template, as I said before, the half ellipsoid, because we found the T cell are very similar to a half ellipsoid. So this is the reason why we do so. And then after we do so, we can move the voxels of the cell to specific location according to the deformation field defined in the diffeomorphic model. And after that, we also normalize the intensity so that we can make the cells comparable. And yeah, because I have talked about the Lvds algorithm, so I will just skip it. So here is a summary of our analysis. So we analyzed more than 16,000 cell pairs. This is the large scale problem, and we have two conditions. First, tumulus, which is a normal condition, B7 blockade, which is a blockade condition where the T cells are not activated. So we can compare two conditions. And the 10 proteins, the last condition is not analyzed in the main pipeline. So here is an illustration of our pipeline. So the first 1 and 2 represent this DIC image, this image with fluorescent. So the small cell is the T cell. This is the APC cell. So because we want here, we only interested in the protein signaling pathway in T cell. So we just label the T cell, but so you cannot see the APC cell in the fluorescent image. And the 3 and 4 represent the segmentation. So we can see they have different orientations, but in 5 we do rigid alignment to make the synapse region facing up. And in 7 and 8, they are this template, and this is the uniform shape with the intensity. So here, because as I said, our images are 3D, so here we only show the center slides for visualization purpose. After this, we can get uniform shape for all cells, so we can make their pixel intensity comparable. Now we can build a model from our uniform this CL with uniform shape. Our model is defined as a min map for each protein, each time point, and each condition. We calculate the min map by averaging the CL of the protein in this point, in the time point, in the condition. This is our model for the protein distribution. Yeah, here is an example of the spatial temporal protein model for 3 different proteins for 2 time points. Here, 0 second means just the start of synapse formation because it's a dynamic process. And in 108 second means that it's the later stage where the synapse has formed. So if we compare the same protein for different time points, for example, for the covalent, we can see the protein pattern are quite different. These are the 3D slices from left to right, up to down. And we can see, compare these two, you can see there's some high intensity in the upper, which represents the synapse region. So this means covalent will be accumulated just at the formation of synapse, while at the late stage, it diffused out. And if we compare different proteins at the same time point, we can also find there's quite difference between these 3, the spatial pattern between this protein. There's no such accumulation for MRLC. And there's some accumulation, but not but there's just a neat accumulation for Web2, not that much as covalent. So this example illustrates we can use our model to compare the protein distribution in both time and proteins. This can enable us to study different biological questions. And besides just visualize and compare the proteins, we can do some more deeper application of the model. First, we can use the model, the protein distribution as feature to compare different time points, different proteins. And also we can do enrichment analysis, which is analysis to analyze the accumulation of proteins in specific time points or specific locations. And also we can visualize the dynamics of the proteins. So here is one figure in the paper. So here we compare the protein distribution for the condition and the protein. So here for each condition protein, we use the protein model, the mean model of all time points as a known feature vector, and use the feature vector to do hierarchical clustering. So from this result, we can see for some proteins, they have quite a similar pattern, but for some proteins, they have quite different pattern between the normal condition for stimulus or the blockade condition, especially for wave 2, you can see their patterns are quite different. This suggests wave 2 may change a lot if we change the condition. This suggests it may be a candidate regulator in this process. And we further analyze our hypothesis by enrichment analysis. As we know, the actin and some of its regulators will be recruited to the synapse region, the upper, this the 3D slices. And so we can define the enrichment region that can represent the synapse region. So we do so by averaging all the models for all time points, all proteins, and the 2 conditions. So this is the mean map. And then we define the synapse region, set a threshold at the top 90% of fluorescence in this model. So we can get the region, the yellow in the lower panel. These are the enrichment region of the 3D stack. And then we can define the enrichment as the ratio of the mean intensity within this region and outside of this region. So if there is enrichment in this region, this means the mean intensity in this region will be higher than the region outside of this region. So the ratio will be larger than 1. And we do the analysis for all proteins at all time points for 2 conditions. The left one are the enrichment plots for the 4 stableness condition, and the right one is for the blocked condition. And we see for different proteins, their enrichment are different. But for most of them, they will have a peak at around 0, which is the synapse formation, and then they will go down at a later stage. So we do some detailed comparison of the 2 different conditions for each protein, and we calculate p-values for each time point, and we found for all these 4 proteins, they have a lot of different time points that are significant. That is, in 4 stimulus conditions, they will have a higher enrichment. This means they will have more accumulation, while if we block them, the enrichment will be reduced. So this means these proteins are potential regulators in this process. And with some prior knowledge, we found wave 2 downstream is the upstream regulator for HS1 and ARP3. So we hypothesize that wave 2 and caffeine are 2 main candidate regulators in this process. And we validated their role by selectively activate these 2 regulators, and we want to see if they can promote actin dynamics and synapse formation in the blocked condition. So here we do enrichment analysis for reconstruction. So the blue line is the enrichment for the reconstruction experiment. So we can see they can indeed increase the enrichment for this condition. And we also do some other experiments. Here we measure the length of the synapse to indicate the formation of synapse, and we found the selective activation can restore the formation of synapse. And we also do this by seeing our marker protein net for the synapse, and we can see also when we restore the in the reconstruct region, the experiment, we can also restore the enrichment for net, which means we can have better formation of synapse. So with these experiments, we validate that wave 2 and caffeine are 2 main regulators for actin dynamics in T-cell co-stimulation. And here is another application that we can visualize the spatial temporal protein distribution. So here, yeah, there's something wrong with the movie.
Is this looking down on the face of the synapse?
Yeah, this is the 3D construction of the cell. So we can move.
But where is the synapse?
This here should be synapse.
It's the face.
Yeah, yeah.
Okay.
Yeah, because there's something wrong with the movie, so I cannot show you. Yeah, here we show the protein distribution of 3 proteins, and with different colors, and we do a thresholding of the protein distribution. So these are binary for each protein, and we can show how their intensity changes across time and space for this protein. So here's a small summary for the model. So our model can, it's an automatic pipeline. It can handle large-scale images, and also we can handle the images with no contrast. And our method can handle the cells with different shape, and we can standardize them and analyze their intensity. And the model has a lot of different applications, such as we can compare the protein distributions. We can do enrichment analysis to analyze the protein accumulation pattern or visualize the dynamics. And we use the model analyze the T-cell co-stimulation. And here's another application. So here we use the model as the input to learn the potential regulatory relationships. So given the spatial temporal model, the T-cell models, we can try to determine whether the change of or in a region in one protein can regulate the protein, the region of another protein. And we apply some methods to learn this kind of relationship. It's basically a causal analysis, and we have published some of the results as a preceding paper in ISMB this year. So here is a basic pipeline. So first we use our model as input, and then we do k-means clustering to define the regions, and then we use hierarchical clustering to group some proteins that have similar pattern, and then we apply some methods to learn the model, and then after that we can find some potential relationships, and at last we compare the relationship with ground truth. So first is the definition of the region, because as we said, the model are.
Could I just ask a question first? I assume that the cells were fixed and the fluorescence comes from post-labeling. Post-labeling with fluorescent antibodies.
No, it's live cell imaging.
So you've overexpressed fluorescent proteins
Yes.
in all these cases?
Yes,
So,
and we take a picture of the movies.
So that you realize that can perturb the dynamics when you overexpress.
We don't find in our science our paper for the project, we found the overexpression level is quite low. They are lots. The gene is inserted in the genome, so their expression level is not very high. It's not.
So you've modified the genome to express a fluorescent.
Yes, yes.
You haven't done it from a
Transformation of
transfection.
transformation. Yeah.
Are those results like cell line?
Yes, cell line. Yeah. Yes. So here we have.
Did you insert in the genome randomly, or was it homologous?
I don't know, because the imaging part is done by our collaborator.
Who's that?
It's Dr. Christopher Wolfing in the University of Bristol in UK.
Can you spell his last name?
Wolfing, maybe I can show you.
Back there somewhere.
Yeah. He's an expert for the T-cell, the immunology, I think.
Where's the research published?
Science-sync learning.
Say again?
Science-sync learning. Science-sync learning.
Oh.
Yeah, let's continue. So, for each model, we put them to a uniform shape, and this template have about 6,600 voxels, which is large if we use each voxel as a region. So we want to reduce the data size, and also we want to define some region with some biological meaning. And then we treat each voxel as an instance and use the intensity for all proteins and all time points as the feature vector. So then we can use these features to perform k-means clustering. And we choose a k equal to 3, and we can define each cluster as a region. So here is our result. So our results this result is quite interesting because each cluster can represent the different parts in the cell. So the region 1, the blue region, it can approximately represent the synapse region. Region 2 approximately represents the nuclear, and the region 3, the orange one, represents the cytosol. And then for each region, we can define the value, or you can consider it as the intensity, as the mean intensity by averaging the voxels in each region. So for each protein, each time point, we have 3 values because we have 3. And then there's another problem that we found some proteins have very similar spatial temporal pattern. This may be caused by the co-localization of proteins in the whole process, because if they are co-localized, they are interact with each other, then they will have the same spatial temporal pattern. And this in statistics, this is called a collinear effect, that the 2 variables, they have an almost linear relationship. And this will reduce the power for our inference. If A and B are collinear, then we cannot know whether A caused B or B caused A. So we want to get rid of this effect. And our solution is to group these proteins as one group. We do so by, for each protein, we can we use the value of 3 regions for all time points as. Finish your thought. Okay. Yeah. For each protein, we use the value of 3 regions and all time points as a feature vector, and we perform hierarchical clustering. And then here is the pattern for the clustering. So we can see some proteins, their patterns are quite similar. And here we do a cutoff here. So we group these 4 proteins as one group, this acting another one, ARP3 and H1 as a third group. And this last 5 proteins, because their patterns are quite different, so we use them as single groups. So now we have 8 groups for these 12 proteins. And for each group, we use the mean value for the regions for the proteins in this group as the value for the group. So now we reduce our system as 8 groups, and each group have 3 values, 3 regions. And then
going back, I guess how is the threshold decided, and
yeah, we tried different threshold. Either we can make a cutoff here, or we can group these 5 protein as one group, and or this 8 protein as one group. And yeah, we show different show the results in our paper, and we also compare the results for different thresholds.
And I guess given that there's a collinear effect, would it make samples instead of grouping it into one sample itself. That makes sense. As in, instead of clustering it into one protein group, would it make sense to simply reduce the effect of individual proteins but keep them separate?
As I said, they have some collinear relationship. It's hard to, if there's some collinear factor variables there, it will reduce the power, because if A and B collinear, we don't know whether A causes B or B causes A. So we cannot infer their causal relationship.
But if you divide your regions into more like areas, not just three,
Ye-
that get rid of some collinearity regions, where if you maybe increase the temporal or double triple, you mentioned that.
So there's two things. First, yeah, as I said, we want to define some regions that have some biological meaning. So for this region, they can represent some some compartments in the cell. And another thing is that if we use more region, it will increase the complexity of the data. Because here we only have 3 regions for each protein. If we have 8 proteins, this is only 24 variables. If we have 100 regions, then we have 2,400 regions. This is a very large scale problem. And for the temporal resolution, because we use our existing data and it's decided by our collaborator, we cannot increase it. Yeah. And also for taking the movies of for live cell, it's very hard to increase temporal resolution because there are 3 stacks. You need to take all the stacks before you move to the next time point. It takes long time. Yeah, this is maybe I can suggest my collaborator if they have money, we can do that. Yeah, so after we clustered and defined the groups, then we fitted some models. One model is called a graph process model, where it defines the system as a graph. So here, for our system, our graph is for each protein and each region. Each protein, each region is a node in this graph, and we want to see how the connection between the protein region in this system. And then in this method, it infers the causal relationship through a regularized multivariate autoregressive model.
Can I just ask the causal relationship here is defined as one thing peaks before another?
Yes, this is, so for causal relationship, there are 2 main characters. One is the cause persists the effect. Another one is if you change the cause, there's change, yes. And if we just get some data, we cannot change the system, right? If we just collect some data. If we do not intentionally to change some thing in the system, so we can only use the first character, just the cause persists effect. So this is the main idea for the analysis, yeah. And another method is called the constrained Elastica net regression method. It has 2 steps. In the first step, we pre-select some causal relationship by Granger causality test. This is a very famous causality test. And then after that, we use this causal relationships as constraints to learn all other relationships. Because this test is a binary test, it will only return whether have cause effect or not. But we want to learn a relationship that have strengths that use some continuous variables to represent how strong the probability of the cause and the effect relationship. So here's some example of using this method. These are the time series. So here the protein dash number, this means the region number. Region 1 means the synapse region. Region 2 is the nuclear, and the region 3 is the cytosol. And the black line is the time series for the cause, and the red dash is for the effect. And we can see there are some obvious lag in the effect, especially for this one. You can see cause first go up and go down, and the effect go up, and at a late time point it go down. So it can identify some kind of this pattern.
Causality means that one really causes another, but you're doing just the time shift correlation.
Yes, sort of. Because as I said, we can, if we do not part-based system, we cannot get the real cause.
This is sort of a statistical definition.
Yeah, yeah. So here we.
See if it's both happening before, if the cause happens before the effect, and if the cause goes down, the effect goes down, then it's much stronger. Yeah. I mean, what else?
Yeah. Yeah, so here we construct our network, and also we want to see the strongest relationship, the time point of at which the relationship happens. That is, if at which time point the cause change a lot, and the effect also change a lot. So here we have the time points. 0 means the formation of synapse, and we can, and the color of the arrows stands for different type of relationship. Black means cause go up, effect go up. Red means cause go down, effect go down, and blue means cause go up, effect go down. So these two are the positive relationship, and this is the negative relationship. So we can see there are some interesting results that the ARP3 in region 1 and also caffeine in region 1 go up. It can promote the actin in region 1, the synapse region go up. This is consistent with our previous results and also biological some literatures. ARP3 and caffeine are the regulators for actin. And there's a more interesting result that in the later stage, as we see, the enrichment will go down. The protein will be diffused after accumulation. We found actin go down in the synapse region, go down. It will promote the diffusion of ARP3 and caffeine. So there's a feedback there. And at last, we compare the two methods, and also with the ground truth. Here, the upper parallel is the ground truth, and the lower parallel are the merged network of the two methods. Here, we don't consider the time points. We just consider whether the top relationship. There's no temporal resolution for this network. And the blue line means the relationship found by both methods. So we can see that for most relationships, it can be identified by both methods, and a lot of them are consistent with the ground truth. So this means the two methods are robust, and also the relationship are robust. And we have some quantities such as AUC to measure the reconstruction error for the network. I didn't show it here. So in sum, for our model, we can handle automatically analyze large-scale images, and also we can handle the images with no resolution. And our model has a lot of different applications, such as compare protein distribution, enrichment analysis, visualization, and also act as inputs for causal modeling. Yeah. And yeah, this is a collaboration of work with the Murphy group and the Wolfing group. And also I need to thank the funding agencies. Yeah, that's all. Do you have any questions?
One of the components was it MRCF? What was it?
MRLC? Yeah, it's the microtubule light chain. Where?
MRLC.
You mean here?
Related.
Yeah. Protein light chain.
Say again?
Microtubule related protein light chain, I think.
Light chain.
Light chain. LC.
Light chain.
Usually.
Ah,
No.
myosin.
Oh, myosin, yeah.
All right, thank you. Yeah.